Welcome. This is Notes from the Lab, an ongoing series of conversations on the
Yeah.
past, present, and future of the
Yeah.
BCG vaccine.
Mhm.
My
Yeah.
name's Denise Faustman, and I am head of the immunobiology labs here at Mass General Hospital in Boston. In this particular podcast series, we're exploring topics related to type 1 diabetes and the BCG vaccine. My guest today is Dr. Singh, who very early on started seeing effects of the BCG vaccine and did some of the pioneering studies of the BCG vaccine as it related to a type 1 diabetes animal model called the NOD mouse. So, welcome, Dr. Singh, to this small podcast series trying to highlight BCG and diabetes. Before we
Mhm.
get into BCG, tell us a little bit about yourself, and maybe you might even remember how you got interested in BCG.
Well, Denise, I came into this being a chemist, learning immunology actually by doing it. So back in early 80s, I was trying to get immune response to insulin. And that's how I got interested in diabetes because, so why insulin? Because, that's the molecule which we lose in type 1 diabetes. So I was interested in immune response to insulin. So I was trying to do induce immune response by using insulin peptides, insulin fragments, and asking how we can get immunity to insulin and understand what goes on in actual destruction of the cells which produce insulin. So that's how I got into it, and it was way before any mouse models came along, and in mid 80s, I got in touch with Ed Leiter at the Jackson Labs. He was the first one to get the mouse model from Japan. And
Mm-hmm.
so I was fortunate enough to set up a collaboration with Ed Leiter to get the mice. So we were the first in Canada to get these NOD mice. But then the question became, how do you study this? So I was trying to immunize mice with insulin peptides in something called adjuvant. Immunologists have been using Freund's adjuvant since almost 100 years now.
Mm-hmm.
When Jules Franz, who actually pioneered the use of using this as a adjuvant. Adjuvant means increasing the immune response. So, so that was the start of it. And what I was surprised that when we took these mice and immunized them with the peptides, and with and without insulin, a couple of months later, I saw the mice were still around. So my technician said, "What's going on? These mice are supposed to have died. Why are they still alive?" And that really started asking the question, what was going on with this BCG? Now, BCG is, historically, it's oil emulsion containing dead microbacteria. So
Mm-hmm.
it was Mycobacterium tuberculosis, MTB, which was the original sort of vaccine for for tuberculosis that was France used back in 1955. So starting that as the basis, so we became really curious about asking what's going on with this BCG, and BCG at the time was not the word used; it was used MTB, M tuberculosis,
Mm-hmm.
because the other variant of the TB in cattle, which is called bovis, Mycobacterium bovis. So it was in cattle. Actually, that version is a milder version, and that actually became the actual vaccine which people used for uh, prevention of tuberculosis. And in fact, it is still the standard of using Mycobacterium bovis for protecting TB. That was the basis of why I got into is asking the question, why the mice which just got the Mycobacterium bovis or dead Mycobacterium tuberculosis is still alive. So that's the basis of why I got into it, and how that is back in 1980, 89, around that time. So our first paper was to actually show that it was strongly protective. And
Mm-hmm.
these mice, once you give them BCG, young mice, 4 weeks, 5 weeks, they never develop diabetes. That's the basis for
They
why.
like it. They like it. So I have a
Well, yeah,
question.
I mean, it became like a big curiosity, which became means asking the question in real science, mechanistically, what's going on? Because to us, it wasn't that mice got cured. I mean, that was a great thing that mice got cured. But these mice are, very disease prone. So non-obese diabetic mouse discovered back in Japan was the genetic base, basis of using um, a model for type 1 diabetes. So
Mhm.
these mice lose their pancreatic beta cells, they become diabetic and die. So
They
so
die.
that's the basis for why we got into it. And
I have
the
a question
old,
for you. Did you ever
yeah,
see a difference between BCG and incomplete Freud's adjuvant or complete Freud's adjuvant? Because many of the original studies were using
yes, yeah.
incomplete Freud's adjuvant. Did you pick up any differences between that and TB or that and BCG?
I'm glad you asked that question because always, you have to use a control. So IFA, the incomplete adjuvant, which does not have the bacteria, is the actual model that you ask what happens with IFA, just oil emulsion. There was some effect, but there was nothing like curing disease. These mice will eventually
Mm-hmm.
become diabetic and they will die. Whereas if you give them MTB or BCG, BCG is just another variant of, that's the cattle variant of the Mycobacterium bacteria, so
Tuberculosis.
Mycobacterium bovis.
Yeah, yeah.
So the BCG is the standard, the
On
vaccine.
the
So
so
yeah, absolutely, there is a
they,
big
oh,
difference between the mice which
yeah.
receive just the adjuvant means of the oil. So the incomplete adjuvant is just simply a oil emulsion,
Hmm.
whereas the dead bacteria or bacterial cell wall is the vaccine.
Yeah, because early on, people didn't really have access to go to pharmacies and get BCG. So many people early on just use complete Freud's adjuvant, which
Yes,
is really a different organism. So I was always curious in those old
yeah,
experiments whether
yeah.
BCG itself looked different than the organisms that made up complete Freud's adjuvant. In
Yes,
your,
I think they are different, because the conventional CFA, complete France adjuvant, contains
yeah,
the Mycobacterium tuberculosis,
yeah,
dead bacteria or freeze-dried bacteria. Whereas the live vaccine, which is used, BCG is a live,
mhm,
Mycobacterium bovis is a live bacteria. So it's not pathogenic in human. I mean, it causes mild disease in animals,
mhm,
but BCG became a standard therapy for cancer therapies and so on and so forth. So yes, uh, live bacteria is the BCG, dead bacteria is Mycobacterium
yeah,
tuberculosis. Yeah.
went
You
on.
don't want to give them people, the bacteria which causes TB.
Yeah. Well, everybody's always afraid of that, right? Everybody's
Yeah,
afraid. So here was an interesting thing. So during those days, everybody was doing mouse experiments. And during those days, you remember Ed Leiter and others, and they put up this list of 1,000 compounds you
yes.
could inject into an NOD mouse, and they all cured the NOD mouse. And when that data started appearing, I felt there was a lot of pushback from
Yes.
using the NOD mouse.
Yeah.
How did you view that data at that time? How did you process that data?
Well, as long as you use the right controls in your experiment, it's not simply to give them any bacteria or any antigen, but if you are careful and carefully monitor the animals, that you keep them in a germ-free environment, the problem is any bug will change the immune response. So you have to be careful that you use clean colonies, you are a pathogen-free environment. So you are not doing experiments simply just for the sake of doing experiment. So you have to be critically looking at what kind of facilities you use, what kind of animals you use, how you keep them, how you uh, feed them, so on and so forth. So as long as you are carefully monitoring, then not anything can prevent disease.
Yeah.
It has to be carefully done. So now there are hundreds of compounds, you can inject, but the problem is that whether we go back those years or even now, immune system is very complex. You change one thing, and don't forget cytokines, the T helper cells, the B cells, macro. There's so many cell types in the immune system that any of them can be, triggered, and they might change the outcome of response. So if you start looking at mechanistically, it is very critical that you simply just don't inject it and say something will happen. Yeah, something will happen. But the problem is, can we experimentally prove that they are immune related phenomena, that these are helper T cells, cytotoxic T cells, B cells, macrophages, dendritic cells, innate immunity? You have so many variations.
So many things going
Immune
on.
system is a very complex business, and if you are not careful, you can you can simply say, yeah, anything will cure. Yeah, sure,
Yeah.
because we don't understand.
Yeah.
So, and I can tell you even today, all these years later, do we know the mechanism of how these things all work? No.
Yeah.
I think we have made lot of progress in gene cloning and finding the right type of responses. Yes, but if unless you analyze carefully, you can get any answer you want.
Yeah. I also think in the diabetes field, everybody started using mice. Everybody started saying everything cured the mouse. But nobody really, or maybe people did the tough experiment and then didn't publish it. But as soon as somebody took an already diabetic mouse with one of those thousands of therapies and gave
Yeah,
compound X to the diabetic mouse and
yeah.
it didn't work, nobody published it. So,
No, no.
nobody publishes negative data. So I think it took a long time for people to get their heads around the concept that for most people trials, we're treating people who have the disease. We're not treating people who are 5 weeks old or the equivalent of a
Exactly.
year old. And when you really started putting to the test all these compounds that cured to diabetic or even mildly
Yeah,
diabetic animals,
yeah,
they all
yeah,
failed. And they all
yeah.
failed except for two. One that had a little bit of traction was anti CD3, but the blood
Yeah,
sugar couldn't be elevated. They just had to be like
yeah,
12 weeks of age.
yeah.
But the one
Well,
that just kept coming through in your hands and
yeah,
my hands time and time
yeah,
again in
yeah.
already diabetic mice was BCG. But by
Well,
then,
BCG, yeah,
yeah.
because it's another model, in terms of the disease, BB rat. There's another the rat model,
Oh, yeah, BB
similar,
rat.
same results, and we found that you can use the rat model or the mouse model, and they both seem to have the effect. So it's not just a NOD mouse, but BB rat is also another
Yeah,
model.
yeah. That's a good
Yeah,
point. That's a good point
yeah.
because
So it's just the mouse and the rat. They both have disease, similar mechanistical.
mhm.
That's how the disease progresses. But as soon as the clinical trial started, I don't know whether you remember Dr. Kevin Lafferty, from
Oh, of
Australia.
course. Of course.
Kevin was the first one who jumped the gun. I think he was in Colorado at the time, and he did some small clinical trial. He gave the BCG to kids, and yeah, there was some effect, but not sure. It was a very
Yeah.
small trial, and and after that, there were so many other, and even I looked at this morning, paper just came out, must be a month ago, about the review on, BCG and disease, and hundreds of reviews come together, clinical
Yeah.
trials, you name it.
But
But
you know what? trials are really hard to do. And I remember that era when people finally said, okay, we got some mice and it looks good. We're going to do human clinical trials. And there's a lot of things. So there was the data also from Israel. And
Yes, yes,
Kevin Lafferty was on that paper.
yeah,
And he
yeah,
goes, I'm going to find 9 new onset diabetics. And
yeah,
I'm just going to do open label, or maybe it was 7.
yeah,
And I'm going to give them BCG. And he gave them
yeah,
2 vaccines of an
yeah,
unusual strain.
yeah.
And he published it in Lancet. He goes, I think I got
Lancet, yeah,
something.
I
And
remember
then
that.
and then everybody ran out, and there were 3 randomized clinical trials after that. There was
Yes,
one
yes,
in
yes,
France, and one in Canada, and one in
yeah,
the US. And
yeah,
nobody looked at the data, and they ran out and grabbed Tice BCG. And this brings
yeah,
up a really important point. They grabbed Tice BCG because it was a bladder formula and said,
yeah,
okay, we're going to put this one to rest, or it's going to be
yeah,
true. And sure
yeah,
enough,
yeah,
3 of 3 trials said this doesn't work. And
yeah,
I look back at that, and the translation wasn't as good as the way we translate now. But
yeah,
nobody had even done a single mouse with Tice BCG. And if
yeah, no,
anybody had even done a single mouse with Tice BCG, it would
yeah,
not have worked. And
yeah,
you think
there,
that it doesn't work in the mouse. And it brings up the important point about strain differences. And you might have even seen this data from the COVID. There was a group, actually up in Canada, but I'm not picking on Canada here, who got Tice,
yeah,
immunized mice, exposed them to COVID, and said
yeah,
BCG doesn't work for infectious diseases. And then
yes,
other mouse people out at Stanford and in Denmark did the same experiment, but they used different strains of BCG, and then
yeah,
it worked really, really well. So
yeah,
I think the era of BCG had to go through a real correction to realize BCG off the shelf of one strain may be totally different than BCG off the shelf of a different strain. And that
yeah.
really put the field back 10, 15 years of
Well,
somebody
not only
tried.
just diabetes, but field overall, was put
Oh,
back because BCG. Don't forget, BCG is clinically used for cancer therapy and
yeah,
bladder cancer and so on and so forth.
yeah,
The problem is the companies which manufacture these, they don't want to make it anymore. Like in Canada, we can't
yeah.
even get the companies. Connaught Laboratories used to make it, but they
Yeah,
stopped making.
Sanofi. And they went
Yeah,
down. They said, we're not interested.
so basically, there's nothing you can use. You're just guessing what you're using.
Yeah, yeah, or what's available, or what's
Yeah,
available.
yeah,
But,
yeah,
the shortages were pretty obvious because anybody who does manufacturing know you can't manufacture the same way you did 50 years ago.
yeah,
So nobody wants to update the plant because then the FDA's there and you
yeah.
got to put another 25, 50 million into the plant. And you saw that happen in Canada, right?
Only everywhere
Sanofi.
is the same problem, and so nobody is really. I mean, I think that for tuberculosis, I was reading this article from Russia. They still give children, the tuberculosis vaccine early childhood
Yeah.
to prevent the tuberculosis. So there are still countries using the BCG, but nobody is really clear what they are using. They won't
Well,
give you
yeah.
the details. So if you really want to do it, you have to be proactively doing it or working very closely with the manufacturers to
Yeah.
see what
To
they want to
understand it and sort it
use.
out. Yeah.
I'm sure
In
Boston is no different.
no
You,
matter how good the data is, as you
yeah,
probably know, there's data out in multiple sclerosis, there's
yeah,
data out in
yeah,
type 1
yeah,
diabetes, there's a
yeah,
lot of data out in select
yeah.
types of infectious
Yeah.
diseases, broad infectious.
My interest, Denise, after we found this was to do the mechanistic aspect of it. So we did all the T regulatory cell, TH1, TH2, you name it, all kinds of mechanistic responses. And we thought, well, there are not people doing the clinical trials, so we wouldn't get into this. We will simply ask basic questions about what kind of immune responses are altered, change.
Mhm.
And so that's what has been the focus since one more thing I just wanted to mention that back when we were doing our OD mouse model, the, the most interesting thing we found was that if you have a fully diabetic mouse and if you do islet transplant, so don't forget islet transplant is a good treatment to reverse the disease. So we found that if you transplant islets in a fully diabetic mouse and treat them with BCG or incomplete France adjuvant,
Mhm.
they were protected from disease. To me, that was the most interesting thing, that the ongoing disease in a fully diabetic animal can be reversed if as long as you give them the BCG at the time of
Yeah,
transplant.
yeah,
If
yeah.
you don't
It doesn't
transplant
work.
them,
I think I think even to today, our experiments and your experiments with
yeah,
are showing regeneration, you're showing transplant,
yeah,
still
yeah,
the only models
yeah,
where you can quote get rid of the autoimmunity.
yeah,
You know, I mean, with anti CD3, you don't get rid of the autoimmunity. It's still
yeah,
there, right? It's
yeah.
still there to some level. It still has those massive surrounds around the islets and stuff. So I'm
Yeah,
still working on, you probably aren't surprised, but I'm still working on BCG.
no,
We have
yeah,
over five, as of today, 550 patients, mostly children, now enrolled.
yeah, yeah,
So these
yeah,
are just absolutely massive clinical trials going on now in the US. And the publications we've done six clinical trials, some of them
yeah,
open label so far.
yeah, yeah, yeah,
Another
yeah.
blinded one coming along. But we think BCG is pretty good stuff. We should change the name for sure. Okay, we should rebrand it, Dr. Singh. Okay, it's
There'll
yeah,
be
you got to give it a new name. Okay, I don't care what we call it, but we got to give it a new name. But you'll be really pleased. The data just keeps coming. The data just keeps coming in humans
yeah,
now.
because don't forget the immune, all this controversy you in the last couple of weeks, all the political thing
Yeah.
about vaccines and autism and you name it, the vaccine is getting bad name. And
Well, I mean, you have to remember when you give it to people with already have diabetes, it's an immunotherapy. It's not a vaccine anymore. So that solves
absolutely,
that problem.
yeah, yeah. So it's immunotherapy is probably the
It
better
is.
word than
You're treating existing
vaccination.
disease. Yeah. It's only a problem when we go into pre-diabetic children, right?
Well, yeah, I mean, and predicting diabetes is still, despite all the progress in the antibodies and genetics and so on,
Mhm.
you're still not absolutely sure that you can predict who will get the disease.
Yeah, yeah.
It
Well,
is
it's true. Even
and
today.
severity of the disease and how much you can manipulate the disease and would they let you do it? That's it.
Yeah, yeah. Well, it's a long road, but I've always enjoyed over the years interacting with you, and of course interacting
Yeah,
with Kevin Lafferty.
yeah,
And all I can say is the data is broad and
yeah.
from many different centers now for these off-target effects of BCG. So you should be smiling for where mouse went to human. It doesn't happen very often, but it's a good story right now. Okay.
Yeah,
So,
no, and the CD3, with Lucien said, no, all the French trials, and they all have done these things, but still, mechanistically, we still don't understand it completely.
yeah, yeah, even the simplest things are hard to understand. But we're so glad to have you today. And from notes from the lab, thank you
Yeah,
for joining us today. And
yeah,
we'll be back in touch, and hopefully, a little bit of history will help a lot of people to
yeah.
understand how this all grew up.
Yeah.
Okay.
No, I'm glad you're doing it. And as I said, I'll send you that article on AI. It has some references related into that, but it'll give you a perspective on, all the future of AI and immune regulation. So this may be an adjunct to your
Yeah, we'll
thinking
see
of
if
how
we can
things
post
working.
them with the podcast.
Yeah,
Okay.
it's a
Well, thank
small,
you.
small.
Thank you very much for the
Thank you,
podcast.
Denise.